Okay, everybody, I think we're recording. Hello and welcome to American Society of Enology and Viticulture Eastern Section. We call this session Hangtime, Grapevines to Wines. It used to be a lunch hour, but anymore it's maybe not lunch where you are. But this is an opportunity for those, if you're not exposed to ASEV Eastern Section, check us out, go to our website, pretty easy to find. And learn about what we do. It's a great organization. It's not just academic. It is for private industry, other industries to be involved and to learn what goes on nationwide. And we say that there is a national ASCV, but this is the Eastern section. So it's pretty much kind of a lot of things east of California. So it is a large part of the country, but extends all the way up to Canada and to New York, down to Florida and all across. So what we found is these sessions have been for broad topics across the country that we can all relate to, but this one is a little bit more narrow focused this month for cold grape tolerant varieties. We're going to learn about some of the breeding efforts that are going on, some of the progress, and just kind of be an industry update as a whole. But before we get started, I'm going to do some introductions and some housekeeping. So my name is Nathan Eason. I work with the University of Georgia. I serve as a county extension agent in the northeast part of the state where we have the vineyard industry is present, and I'm active in that. And I'll turn it over to our panelists today. We have two panelists, and Matthew, why don't you take us away?
Sure. Well, good morning or afternoon or wherever, it depends on where you're at, I suppose. I'm Matt Clark. I'm a associate professor in the Department of Horticultural Science at the University of Minnesota. And for the last 8 years, I've been leading the grape breeding program. And until very recently, that was my title. I'm switching positions and taking over the fruit breeding project, which is basically all fruits but grapes. But I'm here today to talk about some of our efforts to date and where our industry is at developing cold hardy grapes, cold for Minnesota, which have been adopted, really across our region, the northern part of the US, and with grapes and testing, basically from Oregon to Maine and into Canada, and have really been part of establishing an industry where grapes historically couldn't be grown, or Vitis vinifera grapes couldn't be grown. And so I'll kind of lend my expertise and ask some questions of the other panelists as we go along about where we're at and how we study cold hardiness.
Thank you. Jason.
Yeah, good morning, everybody. So my name is Jason Londo. I'm an associate professor also in horticulture, but at Cornell University. And I lead the fruit physiology and climate adaptation program here at the Agrotech campus in Geneva, New York. I've been here now coming up on 2 years, but prior to that, for the previous 10 years, I worked for the USDA as a geneticist, looking into the genes and physiology of cold adaptation in grapes. And so my program focuses 50% on grapes and 50% on apples, and from a very physiology point of view. And so what's the biology of the vine? How is it responding to the climate? Not so much from the viticulture angle, but hopefully the work that we do can help inform breeding efforts, which then can be incorporated into viticultural management techniques.
Thank you, gentlemen. All right, before we get started, I want to do a few things. First of all, if our participants and attendees can go in the chat box and put in the state that you represent, the state that which you live in, and that helps our panelists kind of give us a little bit of feedback on who their audience is. So if y'all can do that now, while y'all enter in the states that you're from, I just want to go over a little housekeeping. This is this session is interactive. It's very laid back conversational. So if you have questions, we just encourage you to, there's a raise your hand icon down at the bottom of your screen. When you want to have a question for our panelists, raise that hand. Everybody is muted. And we can unmute you and you can verbally ask it, or there at the bottom of your screen, there's a Q&A chat box. And we just ask that you type your question in there, and I'll keep a monitor of that chat box and try to get your questions answered. This is interactive. The point is for you to talk to our panelists, not for them just to sit through a presentation. So keep that in mind. Raise your hand or type it in the Q&A. I'll keep a poll on that. All right, so we got some responses, and if I can minimize my participant list here. Oklahoma, Ohio, New York, Finger Lakes, Middle Georgia. We're a little south there, but we're glad you're here, Georgia. Minnesota, Iowa, North Georgia. So, all right, so I got some Georgia folks here. Yeah, so good deal. Thank y'all. And this will be posted, by the way, if you're on YouTube, ASEV-ES. Here in a couple of days, I'll have it posted, and you can share it with whomever you want to. With that, we'll get started. So our icebreaker question is, what is cold hardiness? Matthew and Jason, when we think of cold hardy, I mean, we got some Georgia folks on here that tuned in and said, hey, we're in North Georgia. You know, that's cold hardy, but let's put it in perspective from a 60,000 foot elevation view. You know, when we speak of cold hardiness, you know, we're thinking of acclimation, deacclimation, winter, extreme winter temperatures. And what is extreme? Is that North Georgia winter extreme, or is that North America extreme? And so y'all take that question away, kind of talk to our audience about what do we define cold hardiness as?
You want me to go?
Sure. Yeah. Start us off.
Well, you bring up a really interesting angle on this. The idea that cold hardiness is just one thing or another doesn't really apply. So cold hardiness in North Georgia and cold hardiness in New York functions on the same basic concept that the vine survives freezing temperatures. When we talk about cold hardiness, we're not really talking about chill temperatures, so above freezing. We're talking about below freezing temperatures. The vines themselves monitor the temperature changes in fall as it enters winters in each region, and will uniquely to that region acquire a certain level of defense. So if you take the same variety and put it in North Georgia as you put in New York, the cold hardiness of that variety will be deeper in New York. And that's because the vine responds to the actual temperatures. Similarly, if you take a really mild winter and put that in New York, you'll have less cold hardiness than in a cold winter. And so it's a very dynamic trait, all centered on the idea that you are trying to prevent damage from freezing.
On the flip side of that, Jason, what if you plant what y'all would plant up in New York or Minnesota? What would happen if you planted a cold hardy tolerant variety that y'all use in a southern state that we are colder than the rest of the state? What would that do?
It would probably do fine as far as a growth point of view. It would tolerate your winter. If we were to measure how much winter it could actually stand, it would be less. It would be a warmer temperature, right? So if we take one of Matt's cold hardy hybrids, let's just say Marquette. Marquette in the Finger Lakes will reach down to around -29 Celsius. Hardiness, so it can survive temperatures down to -29 Celsius. And I need to look up I don't actually know what that is in Fahrenheit. My calculator's moving, so it's cold. If you take Marquette, which can survive to -29 in New York, and you put it in Georgia and you measure, it probably wouldn't survive a sudden snap to -29. It would probably survive something like -17. And that's because what I mentioned before, the dynamic nature, the ability to resist freezing depends on the sequence of temperatures leading up to the freezing event, if that makes sense.
It does. And sometimes we can be in Georgia still warm and not ever put that vine maybe in complete dormancy, and that's an issue. We have that acclimation and deacclimation period may not be as strong as it would be in North America.
Yeah, the southern states are going to have less of a strong signaling cascade to give you really deep cold hardiness because you don't need it, right? The vines are not going to achieve a level of cold hardiness unless they need it. And I know that anthropomorphizes it a little bit, like they understand what they're going through, but when you boil it down to the physiological mechanism, the temperatures, the change in photoperiod, the signals that the vine receives tells it what it needs to do because of thousands and millions of years of evolution to say when we reach this point in the season, you need to go dormant, you need to dehydrate, you need to get ready for freezing temperatures. But the amount of that response is metered by the signals that are going in.
Sounds good. Matthew, what's we could add to that? So when we think about the temperatures and that acclimation, deacclimation, what are some of those parameters that y'all are evaluating in your program?
Yeah, I just want to take a step back because we have, I think, two perspectives when we talk about cold hardiness. One is what do the plants do? And Jason's the expert there. And then the other side is the human side. And we have different perspectives of what that cold hardiness is. And most of us rely on the USDA to give us our hardiness zones, and that's mapped out, which is some arbitrary benchmarks about what our cold temperatures are that are averaged over, gosh, I don't even know what the rules are, 15 years or something. And then it gets updated so often. Thanks to climate change, we've seen those hardiness zones shift around. So we all bring our own perspective of where we're from and where we're trying to grow plants and what cold hardiness means. So that's just to go back to the folks in Georgia are in a USDA zone that's different than what we have in Minnesota. And just to bring some context to the whole conversation, where I'm at today at our research center, where we've been developing these cold hardy hybrids, we're a USDA zone 4 and kind of firmly planted in that. And our temperatures routinely get between minus 25 to minus 35 degrees Celsius, which is cold. And it's about as cold as where people ought to be growing grapes anywhere in the world. And then anything else are higher numbers, USDA zones. The numbers go higher as you go further south. And I just bring that up because that's where our target has always been in our breeding program, is that we breed for Minnesota sort of first, which is zone 4. And if plants can survive here in zone 4, they are going to do likely fine in zone 5 and zone 6. As those numbers get higher, we have additional challenges, which are, do the plants have enough cold temperatures, chilling hours, to actually go into dormancy and stay dormant? And then the other issues are it gets hot, right? In other places, hot and humid, and can the crops actually ripen and produce a fruit at a right time? So it's always important to choose the right plant for the right place, and grapes are no different. And the USDA hardiness zones are one of our clues about can the plant survive those cold temperatures? And then other things throughout the season that we're not even going to talk about today are growing degree day units that are accumulated during the growing season and other weather conditions. But when we think about cold hardiness, to me, we have to be thinking about what signals are the plants receiving to help them prepare for winter. And Jason's kind of started with one lane of thought, which is in fall, we have the plants going through acclimation. And that occurs because of probably two key things. One is shortening days, right? So our day length changes and plants are perceiving that. And our plants and that are the grapes that are native in the northern parts of the country experience a greater change through evolution to that time change, right? So we have very short days right now going into December, and those plants, when they have leaves on them at least, are able to perceive that change from mid-June, where we have very long days, to the short days that we have now. That's one signal that those plants are perceiving. And then the second, of course, is that change of temperature. And not just the daily temperature, but Jason's research is leaning more into the oscillating temperatures between those warm temperatures in the afternoon to those drastic changes of cold in the nighttime that really indicate to those plants, hey, we're in fall now, and we're going to be experiencing pretty cold temperatures. And as Jason said, plants aren't thinking about this, but that's the easiest way to communicate them, is that, oh, I've got the signals, I got to get ready for winter. So I think, Nathan, your question was, so like, what are we measuring in our breeding programs to prepare for that? And at Minnesota, historically, the only thing that we've really done to look at cold hardiness is see how the plants survive in the winter. Like, do they die? Do parts of those plants die? Like, the canes and the buds along those canes and the trunks survive the winter, or do the plants die, like from their shoots out all the way to the ground? Plants can die. And then the other thing is that we actually go and assess those buds in the winter time. And we use a technique called bud chopping to do that, where we select, I believe our studies are 48 buds from the varieties and our selections that we're testing. We bring those inside let the canes warm up, we chop the buds, and then we look to see if the bud's alive or not. And recognizing that those compound buds in grapes have, within a node has several buds. And so we look at the primary bud and the secondary bud to see if they're alive. And that's what our program has been doing for, gosh, probably 35 years without bringing any deep analysis to this. There's no other technique than using our eyes and maybe a microscope to see if the buds survived. So the two things again are, did the plant survive winter? And then in the middle of winter, we can make some assumptions if those buds are alive. That doesn't tell us anything about what happened to the trunk, anything about what happened to the roots, but it's a really good cheap proxy that we can do and any grower can go do to see what kind of damage they have. And that has to happen after a cold event to be able to do that.
Well, my next question is again, what time of year? Because a lot of times you get these polar vortexes in February. So, I mean, we're almost even thinking, well, here in the South, spring start sprunging, in March, but up there it would be different. So what would be a time frame you would say to do some of those bud dissections?
Yeah, so for us in Minnesota, you could do it as soon as you have some really like deep winter. For us lately, our coldest temperatures usually happen in that week between Christmas and New Year's, and then about the third week of January. Usually we've had some warming up and then the temperatures crash again, and then maybe again in February. And Jason can talk a little bit about why we might sample at different time points. But for us, like if you're really worried after a couple of days of cold, like really cold, minus 25 Celsius or colder in our region, we might go out and sample some buds just to see what happened, see how those plants survived. A couple of key things if you're doing this at home, and we have some links that maybe we can share when we post this about how to do the technique, is that the tissue actually needs to warm up so that it can, you're not sampling it frozen and being misled. Like when tissue dies, it oxidizes and turns brown. And if it's not warmed up overnight, for example, you're never going to have that death. You're not going to be able to perceive that death to those tissues. So we might sample after a cold event in December or January, and then we won't do it again until later, usually mid-February. And then you get the accumulation of all those cold events and cold injury through the winter at that point. And the reason that we do it at that time is also a good time for growers is because that's when they're most likely to be starting to prune. So if they need to shift their pruning to accommodate the number of buds that have been injured, that's a good time to do that assessment. But Jason can talk about maybe why different types of the winter are better for doing different analyses.
Yeah, I wanted to add on to without the breeder's point of view on the grow outs and the bud cutting. On the more nerdy science side, we use a different technique to assess whether or not the buds can survive a certain freeze event, and by intentionally freezing them to death. And this technology is called differential thermal analysis. And what we do is we go out and we survey weekly through the whole winter here, collect buds from the field, remove them from the cane while they're still alive, and then we put them into these special sample trays and into programmable freezers where we slowly decrease the temperature from 0 Celsius all the way to -40 Celsius. And one of the unique properties of grape is that it has this ability called supercooling. And so the bud itself is able to sort of isolate its vasculature from the cane, right? So it cuts itself off. So it's sort of like a little island. When it does that, it's able to suppress freezing because it gets rid of all sorts of extra water, and it sort of gets really dehydrated and can survive very, very cold temperatures through some tricks of thermodynamics that are not particularly important for anybody to understand, other than it allows the bud to survive very, very cold temperatures. So what we do is we push them over that line. So we intentionally freeze them. And when that little bit of cells at the center of the bud freezes, it lets out a puff of heat, sort of like a dying gasp. And we can register that puff of heat through some electronics. So throughout the whole winter, we can track the temperatures that would kill the buds. So we can see where they are in their ability to resist freezes without needing a polar vortex to cause damage. And just for the listeners, we post all of this data. If you Google Cornell cold hardiness, you'll go right to the website where we post this for grapes. We haven't made it to Georgia yet, but we are covering most of the eastern US. with prediction models based off of this technique. So we can now, we've been doing it enough years that we can now predict cold hardiness given the sequence of temperatures that occur during winter. So invite the listeners to go check it out. Send me a comment, particularly if it's a nice comment. So using this DTA method, we can see what makes certain cultivars better at resisting freeze than others from year to year. And one of the neat things is that with the hybrid varieties that Matt's program has been putting out, that Bruce Reicher's program here in Cornell have been putting out, they use these wild grapevine species that are better adapted to our northern climates. They're better at using that changing day length to gain cold hardiness. So we can see that during the early part of winter, they gain defenses much faster than the European grapes because they're using all the right signals unique to this region, right? So they're much better at gaining quick cold hardiness. Because they gain more early, they tend to be better in midwinter too. It's sort of like they go into overdrive and they're much better at resisting polar vortexes that occur in the middle of winter. When we get to the end of winter, what we call deacclimation, that's basically losing cold hardiness. That process is an initiation towards bud break and growth. And it happens very quickly in response to warm temperatures. The hybrid varieties, again, because they were derived from species that are adapted to living up here in the north, see warming temperatures as an early spring, a need to race to get your fruit out there and ripened before the next winter cycle occurs. So as a result, a lot of these hybrid varieties that incorporate particularly very northern species, they respond in late winter to warm temperatures, much, much more rapidly than the vinifera varieties do. And so the ability of those varieties to survive cold damage sort of flips in late winter, and they become much riskier particularly as we get warmer and warmer springs with later polar vortexes. And so you sort of have this switch off between the two broad groups of grapes in the way that they perceive winter and respond to heat in the spring. And so with this technology, we can see when the safety margin for hybrids flips over into sort of the risk margin. And then we, like I mentioned, we post all this data so growers can go out and see if we get a polar vortex like we did last year. I'm not sure if you guys got the one in Georgia that we got in early February, but we can go and look specifically at that event and say, did it get cold enough to damage the grapes, or were they all more or less safe? And if you look at our last year's data, you'll see that some of the more sensitive vinifera varieties took damage from that because they didn't acquire enough cold hardiness, and that vortex event, I think it was on February 4th, was severe enough, the change in temperature was severe enough that we would expect damage. And so using those techniques in the breeding program are challenging because of the number of samples we need to run for the whole winter, and breeders don't have 15 copies of every selection so that we have plenty and plenty of buds. So our approach to this is to sort of find key points in winter where breeders should be looking at their selections, looking at key points in the acclimation phase to find varieties that respond very quickly to photoperiod, because photoperiod doesn't change, right? As climate is shifting, our falls are getting warmer, but the day length doesn't change. So if we could breed for varieties that strongly use day length as a signal, we can buffer against the effects of warmer and warmer falls. Similarly, on the deacclimation side, if we select for varieties that ignore some of that early heat, they're less efficient at using that heat, we can create varieties that break bud later and hopefully avoid polar vortex events and frost events. And so working together, these two different ways, we're hopefully developing cultivars that have the most resilience to climate weirdness, big shifts in temperature.
Well, Jake, I'm learning stuff just listening to y'all, and I think it's awesome to hear. We actually do have some nerds in the audience because here's a nerdy question. So y'all listen up. All right. It seems that most of the efforts addressing cold hardiness, this is from Alex. It seems that most of the efforts addressing cold hardiness are focused on breeding programs. Certain animals, not sure about plants, express anti-freeze proteins. Is it possible to create a GMO cultivar that would express a similar protein to further protect the vine from freeze damage? I don't know if anybody
Good
has
question.
an answer for that.
I think the question came in maybe before Jason explained the mechanism that grapes use, which is a different physiological and physical process for like extracting water from the bud. But we do know other plants might benefit from this approach that don't do the supercooling. And a lot of plants are good at replacing water within their cell, within the vacuole or between cells with solute concentrations that are higher as one way to reduce or to lower freezing temperatures, right? So adding sugars or salts or something else we all know can change the freezing point of that water. So that's one way that some plants do that. For grapes, I think the question sort of is, can we understand the genes better involved with that process? And that has been one of the bigger sticking points, is that because it's so dynamic and complex, there's a number of genes that are involved with sensing photoperiod, with sensing temperature, with actually driving that whole process of dehydration of those tissues, the development of the periderm that protects the canes and the buds themselves. So there's like all of these things that have to be happening, that it's not a single gene. And for a lot of disease resistance, we're really good at finding resistance genes and kind of this major gene effect. But when it comes to traits like cold hardiness or yield or there's so many different genes that are interacting that it's really hard for breeders and geneticists to figure out what are the suite of genes and what forms of those genes do we need to have, or what alleles do we have to actually get the right combo. I think we've been really successful just through phenotypic characterization, just growing out plants from our breeding programs to find the ones that can survive that cold hardiness, like the deep winter cold hardiness. We've got that. Now, as climate change is occurring, we have better models and better ways about thinking about what's happening throughout this whole winter period. And the paradigm is changing of how we talk about cold hardiness. And it's not just in grapes. I hear it from my colleagues who study any perennial plant, woody herb perennial plant, is that these plants are having to figure out how to monitor the environment. And we've been maybe we selected for the right things to get these industries going and to keep plants alive. And now we have to be really reactive and maybe more aggressive in trying to figure out what happens on the back end. And by that, I mean spring, because we definitely know that those warm-up events are more challenging, and that plants that grow too early in the spring are going to experience frost. They did before climate change. They're certainly going to experience a lot more of those events now. And now we have whole industries based on plants that are quick to grow in the spring because they've used Vitis riparia in their background, for example, which in Minnesota only has 120 days to produce a crop, and they got to grow fast. So I think, to me, like, that's a really challenging part for the plant breeder, is to figure out not just the mechanism, but maybe what genes are involved. And then, do we even have the right plant material that's different enough to be responding differently? And can we measure it correctly? So Jason's group is really helping to set the terms of when do we measure those plants. And great, we can measure cold hardiness in December, but who cares? The plants do that part really well, the ones we selected. It's what's happening in February and March, where we've never even thought about what's going on there. So how do we use these techniques or develop new ones at that, actually, which is now the critical point in those studies.
Well, gentlemen, let's transition to when people are thinking about grapes to grow in these cold environments. We've kind of defined what cold is, cold tolerance. We've talked about how we measure these parameters. But when we think about Vitis vinifera and American hybrid grapes, that's the question, right? What is the threshold? We were talking before we get started, and in Minnesota, y'all have a lot of that lake effect weather. It's a lot colder, a lot of extreme freezes. So y'all are not being successful in growing European grapes without extreme measures of protecting them, insulating them from that cold air through snowfall, straw, laying straw down, burying the canes, And that's a lot of work. And I think what I learned from one of our conferences is it's really not practical from a large scale. So let's just talk about, hybrids, even here in Georgia, hybrids across the country, we're seeing a lot of benefits, but it's also a challenge sometimes to get industry to adapt to those. But with like the success of the breeding program in Minnesota, that Marquette is one of the top grown grapes in y'all state, and a lot of the success of the breeding program up there has allowed that industry to exist. So let's have the kind, gentle debate of hybrids versus vinifera, and how does a grower make that decision?
Sorry, something just fell off the roof. Got me by surprise. In Minnesota, people were trying to grow some of the French American hybrids early on in the start of our industry in the late 1970s, and some of the Vitis vinifera, and it just isn't worth the time and effort. And so we're a little bit different than other parts of the country where we're not up against this vinifera versus hybrid debate, because we don't have an industry if we don't have hybrids. We have pushing 80 wineries in our state who are growing these grapes, producing wines from them, also sourcing grapes and juice from other parts of the country to contribute to what their wine portfolio is. So we have a very different lack of debate in our state about why we grow hybrids. In addition to being cold hardy, that was some of the initial parts of the thrust of our breeding program, but disease resistance is the other key part. Knowing that to be sustainable, whatever that means fewer inputs. If it's economic, it means less impact on the environment, which means fewer sprays. And then also thinking about the impact of communities and societies around that. And hybrid grapes can kind of do all of those things. But the challenge is, how do you market them to folks who don't know those names? And in Minnesota, we're fortunate that our names are recognizable. We named our varieties after places and people that are from Minnesota. So they're things that people have probably pronounced before. If you're from here, Marquette being an explorer, Frontenac, a state park, Itasca, the headwaters of the Mississippi River. And so there are things like that are small hints towards names that people can recognize. But that's really different than what's happening in other parts of our country, where people know 6 to 8 main Vitis vinifera varieties that are grown everywhere else, right? Chardonnay, Sauvignon Blanc, Cabernet Sauvignon, Cabernet Franc. We can list out a handful of grapes and their associated wines that everyone just knows. And so it's really hard to convince marketers that a new product is something that they should pick up. And winemakers, I think, have this great idea of wanting to produce old world styled wines with old world grapes in a place that maybe isn't as hospitable to those varieties as maybe a hybrid would be. So there's a couple of things going on: what the winemaker wants to do, what the grape grower wants to do and can do, and then the marketing that needs to be in place. In Minnesota, we can't grow Chardonnay. So you grow Frontenac Blanc, or you grow Itasca, or our new variety Clarion.
How about in New York, Jason?
We have a much more mixed viticulture here. We have viniferas along the lake shores of the Finger Lakes, down on Long Island, in some places of the Hudson Valley, and then right up against Lake Ontario, Lake Erie, you can grow some viniferas. If you look at a USDA hardiness map, it's basically about the 5B climate region. And it overlaps directly with our AVAs. And so here, if you can grow vinifera, people grow vinifera. And then on the higher elevations or further away from those microclimates is a switch over to hybrids. I think Concord is still the largest hybrid, the Labrascana hybrid that grows in the state, both for juice and for wine. But we have, I think last count, there was something like 60 to 70 different recognized cultivars being grown in the state. And so it's really, really mixed. I would say the challenge of having a mixed region is everyone puts the bar at vinifera, and looks down against I don't like calling them hybrids because they had such a nasty name, but North American adapted or climate adapted, I think is more appropriate. They get looked at as lessers or blenders only or sweet wine only. And I think some of the problems there is that the big 8 that Matt mentioned, or the big 12 vinifera's, there's been thousands and thousands of years of perfection in how to grow those varieties and then make wine from them in a very specific way. And when you grow a new variety, you have to learn, how does it behave in the vineyard? How do you manage it to get the best and most consistent fruit? And then when you get the fruit, how do you take that product and make the best tasting wine? And if it's easier to do tried and true and get a bigger dollar price for it than to spend the time sort of nurturing the new variety and get the best wine out of it so it can compete on the same playing field, you're going to go to this is to my left, you're going to go to the vinifera and try to sell it at a higher bottle price. There are some amazing adapted varieties, climate adapted varieties. The wines that come off of them are excellent because people put the care into making the wine, with the caveat that there are some hybrids that do not make very great wine. And we have enough in the portfolio now that we can work with the ones that give higher consistency, like Marquette. If you go to, like Matt says, in Minnesota, if you go to a state that cannot grow vinifera, you will drink amazing local varieties. They do a great job because they are invested in those varieties and making them the best that they can be. You could do that here too. There is a lot of baggage attached to the vinifera varieties, from sommeliers to winemakers, but up and down, there's a lot of gravity we're fighting against there. Climate shifting, disease pressure shifting is going to push on that baggage. And in some places, I think there's going to be a switch as we move towards more quote unquote sustainable or resilient viticulture. We will have to make that switch. The climate adapted varieties have a ton of potential.
And that's the same way here in the Southeast as well. That's kind of what we're seeing. We still have some purists that are just sticking with the European grapes, and that is great. We support them and encourage growth there in that industry. But it all depends on what the producer's goals are and what their input cost can be. Small grower versus more of a bigger corporate ownership, that those capital costs of spray programs and investment into that vineyard may be completely different from a smaller vineyard who can't sustain such cost. So same thing we're seeing here. And I had somebody flip the script upside down and say, in Europe, everybody's restricted to grow certain grape varieties. You have to harvest it a certain way. You have to process it a certain way. So it is uniform. There are laws behind it. Where here in America, we have the flexibility. It is new ground. It is new territory. Are we going to make some bad wines? Absolutely. But we also have tons of potential to make some great wines. It's just, it's adopting that mindset and working with your winemaker to have fun and to produce good wines. Going through our outline and just looking, what are some limitations to growing regions planting hybrids? Winemakers and marketers like to work with known varieties. Okay, so we kind of covered that real quickly. I'm just trying to go through the topics as we approach the second half of this. So are there other mechanisms that we are exploring in the breeding process that help manage the cold?
Yeah, maybe Jason wants to talk about that. We were in a meeting last week and he was talking about the use of plant hormones and some, really preliminary trials on that.
Yeah, so there's not really other mechanisms from the plant point of view. The primary mechanism for surviving the cold is that supercooling mechanism I talked about. The xylem and the phloem, the vascular tissue in the canes, also use supercooling and are just a tad hardier than the buds themselves. So when we measure buds, we're measuring basically the weakest tissue on the vine. So it's a good proxy for the rest of the vine. We don't understand enough about how to push these vines to their, as if they were experiencing a maximally cold winter every year. We don't know all the buttons to push, but we are starting to learn a little bit about the role of hormones in controlling that acclimation portion I mentioned and the deacclimation portion. So the gaining of cold hardiness in the fall and the loss of it in the spring. And the hormone that we typically are talking about here is ABA, or abscisic acid. And so this hormone is used in lots of different stress signaling: drought response, heat response, some forms of disease response, and cold response, all trigger production of this hormone. And that hormone has just thousands of different things that it can do from the plant physiological point of view. When we apply that hormone to grapevines in the fall, we can sort of make them shut down faster, sort of like overloading that photoperiod and temperature sensor. We convince them that it's a good idea to shut down and go dormant. If we apply that same product in the very late winter, as things are approaching spring, we can sort of apply a brake pedal to the loss of cold hardiness and thus delay how soon bud break is going to happen. ABA is a natural compound you can spray on. One thing we've found out is that it's not very effective in its natural, convenient, and cheap form. If you spray it on the plants, it'll work for a little bit, but the UV light from the sun will break it down really rapidly. So we're working with some synthetic versions of the hormone. So it looks very much like the hormone to the plant, but it can't be broken down quite as fast, and it's much more stable in the light. And that product has sort of the flip side. It's clearly effective. It increases cold hardiness in the early winter and decreases the loss of cold hardiness in the spring, but it is incredibly expensive. And so we have this sort of balancing act in that we know that there's something effective, but the economies just don't line up at this point. We are working with, there's several labs who are working with this product. There's some folks in Ontario, the folks in Pennsylvania. If enough people and enough crops show interest in the application of this product, the economies of scale could potentially bring it down into something that's more applicable for growers in specific frost-prone regions, or areas also where you're getting frost in late fall. That can also affect your cold hardiness acquisition. And so sort of buffering those two sides. That's probably our best tool from a chemical point of view. The other way that we sort of can manage this, particularly in the spring angle, is with double pruning or delayed pruning. So if you leave more cane material on your vines to that early spring, what's going to happen is the apical buds on these really long pieces of cane that you would normally prune off, they're going to break bud. And as soon as they break bud, they send hormones down the cane that suppresses the buds that are below them, right? So if you have early bud break in late February, early March on these big, long, gaggly vines, those breaking buds at the apex, they break, they get frosted off, you come through, you prune that all off, and then your bottom buds flush out. And so it's more of a mechanical mitigation method of getting around climate warming.
And Jason, that kind of goes to, I'm going to RM Kane, I'm going to go back to your question, but Alex asked, early conversation, Matt mentioned sampling for bud survival around the start of pruning. How can information gained from that sampling be used by growers to adapt their pruning efforts? And I think you're kind of answering, you're touching on that. What might have to change depending on what they might find? Now, what we're experiencing in the Southeast is I think everybody across the country has concerns about late freezes. And we've had those the past 2 years. And in our studies, we try to delay that pruning, and we talked about this last month as well. But if you wait too long for that bud push, for those new shoots to start pushing out, and then you come in and do a final prune, if you wait too long, it can inhibit bud push at those basal buds, around those 1 to 2 basal buds that you're really wanting to come out. So there is too much delay, if you want to call it. So I'm all ears to hear how you might want to answer that.
I think to the first question from Alex is, when we do our bud chopping, it gives us a percentage of buds that are damaged. And when you're thinking about pruning, you need to think about how many live buds you have per unit area, whether you're working on per vine or per cordon, per spur, you know, and there's some great, like the Ravad index and others that kind of give you a sense by variety how many buds you should be having per linear foot. And so if they're dead, you've got to think about leaving more of those buds. And so if you have 50% and you want to have 30 buds on that vine, you probably should leave more than 30. Maybe that's 60. And then once those plants start to grow, you can come and do some shoot thinning to get that vine back into balance with what you like if your estimation is off. But if you prune like normal and you had 50% bud kill, you're going to have too few of live buds on those vines, which is setting yourself up to have a lower crop. And then you're going to have these blind spots throughout your canopy. So that's where that number really comes into play, so that you can estimate what your pruning efforts really need to look like to reflect how much injury you have to those buds. And then, you know, if you have complete bud kill from your sampling, then you're gonna have to be thinking about retraining those vines and finding some dormant blind buds that maybe come along the trunk or from the cordon and reestablishing a canopy. So it can be disappointing, right, if you had an extreme weather event and you don't have any live buds. It's a good point for a vineyard manager to be sampling that wood across the vineyard and knowing where those canes are coming from, and to have weather stations or other sensors across the vineyard can be helpful too, because you need to you might have to prune differently across your production area, right? If you have some lower spots that are going to be at greater risk, those might be have a different pruning than someplace else in the vineyard.
Matthew, I presume that that sampling of buds is happening in that lower 3 to 4 inches of those spur points. Is that correct or not? Where are you sampling? What's the protocol on that?
Yeah, Jason can talk about that. I would have made the same assumption that the closer you get to the cordon or the trunk, the more hardiness there is. But Jason's research has shown that it doesn't really work that way, that the buds operate independently. You're nodding your head. Do you want to say more?
Yeah, I think actually the very basal buds are sometimes weaker, but in general, the buds that go into dormancy sooner tend to be a slightly bit hardier, but by the end of winter, they've all caught up. So if you start out in the beginning of winter and say you sampled buds 1 through 20 on a cane, you would see maybe the first 2 buds are a little bit weaker, and then it is more hardy and decreases hardiness as you move towards the tip. By the end of winter, with the exception of like the last 3 buds, right? Because that's the weak wood that barely got into dormancy before the first trees. You should always prune that off. You should never consider that good wood. By the end of winter, they basically all caught up, and they're all about the same cold hardiness.
Yeah, you should really just assess the buds that you're sort of planning to keep. Like Jason just said, if the shoot you're looking at is a diameter less than a pencil, why would you keep that shoot for a cane or a spur anyway, right? That should not be a good assessment, a bud for you to assess. Use the actual tissues in the vines that you're planning to retain, and you're going to prune them out anyway, so you shouldn't feel bad about removing a few samples to get 50, 100 buds out of a 1 acre, 10-acre vineyard. You have a lot of plant material to work with. We don't have that in the breeding program. Jason hinted at that earlier. For some of our earliest selections that we're testing, we have 4 vines, right? So we don't have a lot of shoots or canes, I guess, to be even looking at those buds. And we want to propagate some of those, make cuttings or graft, whatever that the case might be. So we're really pretty limited. That's why we stick to 48 buds. And that's something that we can do pretty quickly with our team to look at.
And is there, again, have y'all made a publication on that sampling protocol?
We have a bud chop workshop that is on YouTube that maybe we can link.
I was just trying to think about how the audience might could get access to that, because that seems very applicable to a private vineyard to learn that technique. So any link that you can provide in the chat box, if you'll post.
I'll look for that. The cool thing is that we used to say, well, you need a microscope, just a small light microscope. But now we have these things, right? We have smartphones that you can zoom in. And so you can see really pretty quickly what that looks like. And I can just show an image real quick for the folks who are on what I'm talking about. Hopefully you're seeing this PowerPoint slide.
Yes.
So the image on the left shows a cross section through a bud, and we can see that at this node, there's major. I don't know if you're seeing my.
Yeah, we can see it.
We're seeing you, Mouse.
Okay,
Yeah.
cool. Where the primary bud is, which is this larger one, is dead. It's been sliced through, and you notice that it's brown. So living tissue in plants is green, especially if it photosynthesizes. So this one has died. We can see a secondary bud here, which will push out and produce some fruit. So that plant isn't a complete loss. And then if you look closely, I think this is a tertiary bud down here, the smaller one that's also alive. So this is what you're looking for. And again, this picture was taken with a microscope, but now that we have smartphones that can zoom really pretty good, you should be able to see these tissues. Take that picture, evaluate it a little bit more, and you can see, do that at home really pretty easily without any special tools. Again, when you're slicing through these buds, you don't want to go too deep, because what you'll be looking at is the vascular tissue as opposed to the meristematic tissue. In the meristem, what we're looking at here, these concentric rings, really are what telescopes out in that early growth and early spring that has leaf initials. So like these preformed little leaves and preformed little flowers. And if those are damaged, you're not going to have any fruit this year. So again, if you cut too high, you're not going to capture where those growth points are. And if you go too low, you're also going to miss it. So you kind of got to perfect where your cuts are coming through. And our video walks through how to do that.
Perfect. Quick question here as we kind of slowly come to an end. Has anyone done research on the impacts of rapid spring warming and limited cold freezing pressure?
Yeah, so I can talk a little bit about that. So one of the things we didn't really cover here is the other aspect of winter, which is chilling. And so they're surviving winter freezing temperatures, and then there is gauging how long winter is so that bud break is timed correctly. And that is, most people are probably familiar with that as chilling hours or chilling requirement. Normally when you would order, if you order from like a Gurney's or something and you order fruit trees, they're going to come with a chilling hour recommendation. And so that it's the amount of time spent at low temperature, but not freezing. So like around 1 or 2 degrees Celsius to around 10 degrees Celsius, or 35 to around 50 degrees. The grapevines are able to sort of measure that amount of time that they spend in there. So every day that it warms up into that area, they start counting. And then at night, the temperature goes below, they stop counting. So the clock stops. If it goes too warm, the clock stops. And then if it goes really, really warm, the clock can actually go in reverse, depending on the model you use. So if you're in a region that has limited cold or freezing pressure, let's say Georgia, you would be assumed to accumulate less chilling than something like up here in New York. We get around 1,500 hours of chilling in New York. And I don't know what Georgia gets, but let's just say 1,200.
Yeah, 1,100.
Okay, so let's make it even more dramatic. Let's say 700 hours, right? So if you grow something in Georgia that is used to a long winter and has a higher chilling requirement, it is not going to respond to Georgia's winter in the same way. It's going to, for all intents and purposes, respond to the winter slower, right? So its perception of winter's time is slower. So you have sort of a built-in buffer against sort of this rapid spring warming event. When you have rapid warm temperatures up here in the Northeast, and the varieties have reached all of their chilling that they need for winter, for their senses to say winter's over, they respond extremely fast to that heat. Like I said before, adaptation-wise, they want to break bud and set fruit and go back into dormancy before the winter comes around. In Georgia and further south, because you have less of those chilling hours, those varieties are sort of going to have a little bit of protection against that rapid response to heat. So this research on the impacts of rapid spring warming in warmer regions, there isn't a lot of research. There certainly isn't a lot of research looking at the cold hardiness profile the way that we do it up here. in part because you don't have the midwinter stress to worry about. But there are people who are starting to look at the phenotypes I've been talking about in the South. So there is a group in Mississippi who are now using that technology. I believe there's actually someone in Georgia, and I don't know their name, who's collaborating with us to look at it. And we're doing a project with Arkansas now to look at cold hardiness response in muscadines and bunch grapes in that more mild climate. And so we'll be able to answer some of those questions, but in general, the warmer winter climate sort of slows down the reaction speed that you would expect of a variety up here. Hopefully that made sense. Sometimes I talk.
Yeah. And, I think what's interesting, what I've learned today is, the modeling that y'all are doing and these markers of extreme stress events correct me if I'm wrong, but like you said, that could be applied to across the country, that technology, that understanding of those physiological stages, those extreme stress events. So that's kind of exciting news.
Yep. And the one thing we need is more data from warmer regions because we don't have the history of concern. We don't have a lot of labs in the South that are checking these phenotypes. And that's really important for the models because the models we build work great in the North. We know, but they're dependent on the chilling models that I just referenced, how we track this winter. There are probably 8 or 10 different chilling models, depending on the fruit crop you use and the region you grow in. Because when you get right down to the nitty gritty, we have no idea what is going on to complete that chill requirement. We just have a history of consistent winters that give us chill models. Now that winters are changing, those models are not necessarily accurate anymore. And we find actually, if we use our models on Long Island, for example, who get double our chilling hours because the Atlantic is so moderate, they get up to 3,000 chilling hours. Our models start to fail pretty successfully in late winter because we do not know how to capture that excessive chill within the model predictions. And the same thing applies for southern regions. We just don't know. We don't have the data to figure out what we're doing wrong. But we're working on it. Like I said, we've got people in Mississippi, Texas, Arkansas, all contributing, hopefully, to make this model better and better.
Well, we have coming on the end of time. Is there any more questions from our audience? If not, and you could type that in while I finish up here. Y'all can look these guys up, send them emails directly if you have questions. The purpose of this is just to expose you to some of the research that's going on, some of the understandings of what we know about certain topics, and to share in the frustrations of what we don't know, but know that we're working as a team together to improve those answers. But we appreciate y'all for hanging on. Matthew, Jason, is there any last words of, one of the things is this breeding, the selection process, it takes time. So sometimes people don't like to hear that. They want Minnesota to find an answer and 2 or 3 years later have something they can buy on the market. But the facts are is this stuff takes time. Y'all been successful at that, but what are the time frames when we think about some of these selections that we're choosing?
Yeah, the breeding process for a new variety can be upwards of 20 to 25 years by the time we go through making the cross and the evaluations. And fortunately, we've been part of a multi-state testing program that Cornell and Missouri and Kansas and Michigan and a number of other states have been part of, which allow us to make some better decisions about what gets advanced to become a new variety. But it, like you said, it takes a lot of time, and we have a huge matrix of traits that we're interested in, and every variety doesn't hit all of those boxes all the time. Clarion, which is our most recent release, excellent wine quality, great for the grower, downy mildew resistant, not cold hardy to zone 4, powdery mildew susceptible, but it has a manageable growth habit that makes it really nice for the grower compared to something like Marquette that is wild and vigorous and puts on lots of growth and lots of laterals. So you know, balancing out this matrix of traits is really hard. And I would say all plant varieties have problems, and sometimes we don't know those until they leave the breeder's hands and they get in growers' hands. But I mean, Jason and I were just in Michigan last week, and we went to a couple sites growing Vitis vinifera, and those plants are problems, right? They have problems on those sites, just different ones than what a new variety might have. And there's some cultural and societal and academic studies of how to work with those plants, but you put them into a new environment and you've created some new problems. So my hope is that we develop products that seem to work well for us here on a limited scale, testing in a few places, and that growers try out new varieties, whether they're these climate adapted varieties with North American species in their backgrounds or Vitis vinifera. But trialing new things to get the right plant in the right place is what all horticulturalists should be doing. We do it with corn, we do it with soybeans, we do it with carrots and squash. Grapes should be no different, and farmers should take some onus to put in some small plantings and see what works for them. And then unfortunately it takes investment, and farming's a big risk, but getting the right variety in the right place at the right time is the best strategy to be a productive farmer.
Well, on that, I think that's a great way to wrap up. That was a good concluding statement. I want to thank our audience again. Encourage you to check out ASEV, American Society of Enology and Viticulture Eastern Section for more information. We'd love to have you as a member. Checking out future Hangtime sessions. Come to our conference. We're going to be in Ohio this coming up summer in 2024. So a lot of good things happening. Jason, thank you for joining us in this conversation. Matthew, again, my contact information is, I didn't put it in the chat box. Anyway, Nathan Eason, UGA Extension, you can find me if you have questions about the recordings or anything like that. With that, I will stop recording. As soon as I figure out, there we go, top left.